Oh, okay. So, here we go. We're marching into DNA. What it is, what it does. We've already done this once, but we're going to do it more because we've got to really get refamiliarized with the concept of nucleic acids. So, let's start straight. I'm going to draw. I promised I would stop drawing these, but I'm a liar. I'm going to draw a beautiful molecule here you've seen before. Okay, so everything that's going on here, this is obviously a carbon. So this is C1, C2, C3, C4, and C5. Stuck to this would be an HO and an H. Two of them, actually. That is a very interesting molecule. We've seen it before. I want you to clearly look at this carbon, carbon number 2, and notice that it has no oxygen. So what we call this whole thing is, this thing is deoxyribose. So this is a 5-carbon sugar, 1, 2, 3, 4, 5, and it has these beautiful, tasty hydroxyl groups that are just waiting to be stuck on something. Got a little bit of a chalk shortage. Look at that, and look at that. So here's our molecule. This is kind of the base kind of understanding. Or excuse me, it's not the base. This is the understanding. This is the beginning of understanding what nucleic acids are. And of course, getting back into that concept of what nitrogenous bases are and phosphate groups. And of course, all three of those things coming together to form nucleotides. So let's move on into more of a cartoonistic version of this. Always remember this. The most important part of this is, of course, that you know that it's a ribose and that it has a 3-carbon and a 5-carbon. These are the ones we're going to follow. So, here we go. So now if we get a little bit more cartoony, we will go like this. And that's how we're going to draw one ribose. And that ribose is going to be stuck to a phosphate group. So this is our deoxyribose. So this is our sugar. And then up here, we'll stick, just in a cartoon, we're just going to stick a base. Now remember our 4 options for this, A, G, C, and T. Those are the 4 options there. And now we can clearly understand what is going on here. This part, I need a better color. Ah, look at that, spectacular. Okay, so this here is going to be what we call the 3' end. Very important, 3'. And then up here, this is going to be the 5'. So this is going to be the basis of us understanding the directionality of nucleic acids. So remember all of that stuff. If you guys wanted to get back into the mix, you'll know that right around the corner is the concept in double-stranded DNA of complementary base pairing. And we go A to T, C to G. You remember that. You might also remember there were 2 categories of bases. We had pyrimidine bases, we had purine bases. You can go right back to your notes and get tight on that if you want. So we're going to go a step further here and take a look then at kind of the whole deal. So here we would have a phosphate. I'm just going to put this together like we're used to seeing it. We always want to make the bend in that shows off of the carbon 5. We always want to make that bend when we do that. It gives us a great appreciation for the directionality. Then of course up here, let's say we have an adenine. Let's see up here we have a guanine. Say up here we have a thymine. So I just wanted to let you know that this little bit of this little mini strand, this is the 3' end, and this is the 5' end. So we have that directionality. And of course, if this was a double-stranded DNA, we would have that complementary base pairing. So upside down, there's a T. Upside down, there's an A. Upside down, let's see, that's a C. Let's see, that's a C. Sorry to do it this way, but of course we got our hydrogen bonds here. And then if we do this thing upside down, this base would come at us like this. And here is our sugar. And here is our phosphate group. And then here comes our 3', 2 of phosphate. And then that, and then here comes the 3' to a phosphate, and like that. Oh my! I think we pulled it off. It's not precise and beautiful, but now we actually have the most important part of that. Is that this thing here is the 5'. I'm sorry to do it upside down. If you're working with a phone, you could flip it upside down and this would look good. And then this end here would be our let's see, 3'. Here's our 3'. So the idea then is, in this concept, is anti-parallel strands held together by hydrogen bonds, double-stranded DNA, and this is a very, very short little snippet of double-stranded DNA. Very important for us to understand this basic business, because what we're going to go into first off is we're going back to this preliminary business that we saw in mitosis and even meiosis. DNA has to be duplicated. What we're going to describe is a dynamic process of unzipping double-stranded DNA by breaking the hydrogen bonds. That's what all these dashes are, right? H bonds. So we're going to break these hydrogen bonds, and of course we're going to expose nucleotides, and we're going to establish nucleotides coming in as units in a complementary fashion, A to T, C to G, to actually build DNA, nucleotide by nucleotide, establishing the sugar phosphate backbone. Oh, I think I'm going to go ahead and put that on there. So we have this is double-stranded, it's complementary base pairing. The base pairing is established by H bonds. Everything else that's indicated here would be a covalent bond. Of course, way back months ago, we identified that hydrogen bonds are weaker than covalent bonds. So if you start tugging on this molecule, which is literally what is going to be happening when we get into the DNA duplication process, to unzip it, we're not going to be breaking the covalent bonds, we're going to be breaking the hydrogen bonds. Okay, it's spectacular stuff. Lots of great things to do. Overwhelming detail in the text. Many beautiful and quite high quality. I kind of applaud some of their diagrams in the text. They're not overdone to the point where you kind of get lost in the artistry. You can see still what's going on. But again, it's overkill. There's so much in there. So my job is to break this down and make it, accessible, and build that scaffold in your brain where this lives. Not all the hyper detail, the big picture stuff, so you really understand it. So that's where we're going. So where we'll start here is we're going to look at DNA replication. Now, a long time ago, we talked about cells, and we had differences between prokaryotes and eukaryotes. Now, I'm more about eukaryotes. I think you are too. But there are complexities that arise in eukaryotes. I'm going to be spending most of our time talking about eukaryotes, but we have to start and discuss what happens in prokaryotes. So you may remember that when it's actually, I think the best way to look at it is when we looked at mitochondria and chloroplasts, we described their nucleic acid, because they had DNA, as circular chromosomes. You can go back and take a snapshot of that, but we were drawing those things in those respective organelles as little circles. And of course we had ribosomes there too, but the nucleic acids, DNA in both cases, were little circles. So that's the way it looks. So if we drew a picture of, this is very, very oversimplified. This is double-stranded DNA, except it's a circular chromosome. It's a circular chromosome, and what we're going to identify is a point here on this circular chromosome that we are going to call the origin of replication. Origin of replication. What we're going to see happen is breaking. So let's make sure we understand this. This is one strand of DNA, and this is one strand of DNA. So these guys are, between the two of them, complementary. And therefore, this circular chromosome is double-stranded DNA. Very important for you to know that. So, if we blew this up, of course, we would see individual bases. We would see, the life of the party would be all these little hydrogen bonds that hold this thing together, which I'm not going to really draw. Just this is a very, very kind of basic cartoon of the process. So here's our origin of replication. And what we're going to see, without identifying any of the machinery that causes this to happen. We'll get into those kinds of things later. We'll just look at the process. So here we have this chromosome that has a bubble now. And what's happening here is, I'm going to use green, I hope it shows up. We're actually going to, I need to always identify these origin points. There's the origins. And what we're going to see happening here is complementary strands forming. So that's complementary stuff that's forming. And this thing just continues to get bigger and bigger and bigger. This bubble starts to look like this. Here's the new stuff. Here's the new stuff. And here's our bubbles. Okay? And then, our origins. Continuing on down the line. eventually you get to something that looks like this. So that's this part right here. And by this time it looks like this. And then finally, you end up with completely brand new, 100% duplicated entities. And then, of course, one of these guys completes the cycle. So there is the beautiful, beautiful cycle of the process that takes place in prokaryotes. So now what I want to do is describe just this general cartoonistic summary of what happens in eukaryotes. So there's prokaryotes. I'm going to erase all that. I hope you have it. It's beautiful. And get circular chromosome out. Get prokaryotes out. Get all of this business out. And here we go. So now, in eukaryotes, we'll look at the process of, here's a chromosome. So, that's one strand of DNA. That's one strand of DNA. Of course, between the two, complementary base pairing. And we got that going on. Of course, I'm not going to draw all these, but it would remind you that, of course, this is held together with hydrogen bonds. And now what we'll see in eukaryotes is several origins. Let's just draw 3. So 3 origins here. So that's what these blue things are. Origins of replication. And of course, there's multiples that I've drawn. And here's what's going to happen then. Over the course of time, we're going to see so we still have our intact, hydrogen bondings, double-stranded bits here. We're going to identify the origins as we did before. They're there, they're there, and then I'll use this green chalk to show you that we've got this going here. Now, technically speaking, this is a lot more dynamic than you might think. This is a grotesque over-cartoonization. We have made this thing, so there's a lot more detail here, and that's what we're going to do next. So this is note. This is a cartoon. We need a closer view. We need more detail, and that's what I'm going to do. We're going to get that more detail. I'm actually going to have a meeting with the 11 o'clock class right now. This is kind of interesting. This is where I'm at in my day. So I'll shut this down and go to the computer, meet people, come back, and I will show you the more detail that I've promised. All right. That was a good meeting. Time to pick up the story. So remember that I said that was very cartoonistic, and now I'm going to show you why. There's a lot more detail here. So we're going to draw a very nice picture. So first off, this process, DNA replication, is enzyme catalyzed. So enzymes are playing a big role in this, and that's important. We'll talk more about that in a minute. The enzymes in particular are called DNA. These are great names. Polymerases. DNA polymerases. Polymerization. Oh, this is awesome. DNA polymerases. And a very important point is that most DNA polymerases are going to require something. Most require what is called a primer. Most is going to require a primer. So there's some new things to throw into our concept now. We're going to see, I'm going to draw a picture and then put some names on the board. So here's going to be a replication bubble. Now this is really important because there's our hydrogen bonds, there's the bubble. I can go ahead and draw our origins. So this is what we saw before. Now we're going to put some real story together. This is the 5' end of this strand, so therefore this is the 3' end. It's complementary, so this is and anti-parallel. So this is the 5' of the lower strand on the board, and this is the 3'. So what we're going to see here is quite spectacular. I'm going to keep with red for a primer. So I'm just going to draw a piece of red here, and then I'm going to draw a kind of a mirrored piece of red there. And then I'm going to draw some small pieces of red, and you're going to see what this means in a minute. So I want you to do that, and then I'm going to draw a big, long, beautiful piece of green and an arrowhead. Big piece of green and an arrowhead. A piece of green here and an arrowhead. A piece of green here and an arrowhead. I'm going to explain all this in a minute, but we've got to get this whole diagram together. You'll definitely see one of these on the final. Okay, with lots of questions. So we got that. Then over here, you have to make sure that you clearly see that these are continuous, but that's red and this is green. Hopefully these colors are showing up well. And then finally our last one is right here. All right. And arrowhead's showing the directionality of it. So if I got yet another color, if that shows up, got a color here. So this is 3' and 5'. So this is all new stuff that's forming. And so if that's 5', this is 3' here. This is 5'. So that little strand is growing this way. Let's label some other bits. These big parts here are called leading strands of new DNA. This is a leading strand. And then these other bits here, these smaller ones here, these are called lagging. These red parts here these are primers. So as I mentioned, DNA polymerase requires a primer. The whole word primer, to prime something, means to start, the beginning. It's the first part. So primer, 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 primer. If you think about this in terms of nucleic acids, something about this primer has to be, it's specific. It's made of RNA. I should have mentioned that. and it's usually 5 to 10 bases long. That's a good question. And these things have to actually be complementary to the parts of this bubble here for this thing to happen. So this is the first thing that happens, and then this is the direction of synthesis. We know that the leading strand on each side is the big long one, and then we have these small lagging strands that are down here. So I'm going to tell that story with a little more detail. First off, let's get a little more detail for the primer. Usually, this thing is 5 to 10 bases in length, and let's see. It's I told you it's made of RNA. So that you've got to remember though, this is RNA, so it is a ribose, not a deoxyribose for its sugar, and then the bases would be adenine, guanine, cytosine, and uracil. Remember that one. So there's our primer. This is the whole story. And let's see if I can throw a few bits of notes on the board to show what this is. I think that we need to get some sort of a concept of DNA polymerase being in action here. I'm going to draw DNA polymerase kind of like a little yellow cloud. Here's our yellow cloud. That's DNA polymerase. Right here. This gives you an idea that this is kind of a sled, something that's kind of cruising along the process here. So this little DNA polymerase is acting right here, where everything is, where everything is happening. Okay, that's that. Okay, so that's a lot of detail, a lot of crazy detail. Let's see, I'm going to call these lagging segments and then give another name to them. So lagging, wish I could do this, segments. And down here, I'm going to say that lagging segments have a special name. These lagging segments are called, also known as Okazaki segments. Or fragments, actually, is what they call them. Okazaki fragments. Okazaki fragments. So we're going to see this process then of the DNA replication phenomenon, where in this particular case, let's write down, if this is 5 prime, 3 prime, this is 5 prime, 3 prime, we're going to see that in every case that the direction of synthesis here is downhill. I'll put that here. I'm just saying downhill because 5 is higher. 5 is higher to 3. This is the direction of synthesis is indicated by the green arrows. And everything that's green is actually new DNA. That's a new DNA strand that's forming, all the green stuff. The primers are still here. So let's visualize this process now that we have all this detail. So in the bubble, this is how it's happening. So we have leading strands that are continuous and long, and they'll get longer and longer and longer. But but behind in each bubble, we have this ever-increasing, growing number of Okazaki fragments. When these guys reach all the way, you're basically you don't have a pure complementary copy of each strand of DNA. You have DNA and RNA, DNA and RNA, DNA and RNA, DNA and RNA. And we've got some work to do. And it's spectacular work. We're going to have to get rid of the RNA and replace it with DNA and fix and stick everything together. So we have that uniform continuity of sugar phosphate backbone. it's going to take a lot of action, and the action is going to be related to a lot of enzymes. So that is a flat out beautiful board. I love this board, and I'd like to tell you more, but I think we need to get more space and tell the rest of the story. Okay, so now what we have to do is we're going to have to take this this is giving you kind of an idea of something that's there. So we got to basically, this thing, this needs to become that through the whole process, basically. And that's a huge deal. So it's going to require some enzymes. Enzymes are going to be required. And here they are. Helicases. Helicases will untwist the double helix. We're going to need something called topoisomerase. Topoisomerase will eventually relieve the twist. We're going to need some other factors. Let me get this. This is DNA polymerases. And here's our DNA polymerase list. We're going to need first DNA poly 3. Why am I putting them in a backwards order? I'll explain in a minute. We're going to need DNA poly 1. They call them poly 3, poly 1, instead of the full name. It's just, I guess, a shortening of the deal. And then we're going to need what's called DNA ligase. And then we're going to need some other factors. We're going to need what is called protein single stranded. Oh, wait. Sorry, we're going to need single stranded binding proteins. It's a lot of stuff. I have no idea if I can get the definitions there. But let's take a look at them. So I think I've told you what helicase does, topoisomerase does. The DNA polymerases collectively are adding nucleotides. And I'm going to say complementary nucleotides. And DNA poly 3, it adds nucleotides in the 5 to 3 direction. Okay, God, this is crazy. It's a lot to get on a board. And DNA poly 1, which I've listed later, it does happen later. This is really important. It replaces, and I'm going to use color coding here, it replaces the RNA of the primer. That's the big part. That's the huge part. Replaces the RNA of the primer with DNA. So this is huge. That's that part. Look, RNA, 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 DNA. We got to replace it. That's there. Ligase is going to be important because it's going to bond the DNA. It'll bond all this DNA together after RNA does its work. After DNA poly 1 does its work. So after DNA poly 1 does the substitution. So DNA ligase is what's going to actually enable, we're taking these out and we're replacing these with these and taking these out, replacing these with these, and then basically DNA ligase is going to take all these arrowheads away because now it's smooth. Are you with me? That is what's going on. A little bit more on these single-stranded binding proteins. I don't know how important that these are, but after the parental strands separate, these bind to the unpaired DNA strands to keep them from repairing. So way up here in the early bit, I'm going to let this be kind of a check it out in the book because this is the big story for us. All right, that's exciting. Oh, did I talk? Yeah, I did talk. Okay. I think one thing I should do about DNA ligase is that it binds, it essentially fuses Okazaki fragments together. There we have it. Okay. So that's a pretty good board. It is a very detailed board. The great thing of it is that you can take this into the textbook and you can just see wealth of information. Again, it goes over the top. Sometimes there's too much information, but you can see that and kind of embellish it to get the story. I think though, if you step back and you say, hey, we were going from what I used to have up here with those little red bits, you know, we've got to go from this to this. Here's the players, the major players. And it boggles my mind because this is happening all the time. We've got to completely duplicate that DNA. In the you remember the cell cycle? Here we had G1, S, G2, and remember what S was? And that's when we're duplicating DNA in the cell cycle. This is what's happening, and when we think about it in the context of the cellular biology, the bigger picture stuff of we're actually going to end up with two nuclei that are clones of the first nucleus in mitosis, and then we had that exciting stuff in the middle where we had the duplicated chromosomes, two chromatids and a centromere. Now you actually put a higher magnification on it. It's crazy. It's mind-boggling. You would think this thing would just go perfectly linear from one end to the next. But you think about the efficiency of this. If these are all going at once, you are, if you were rate limited in just having one arrowhead, all you could do is work on one end. If you have multiple work sites, you can go a lot faster. You know, think about this. We're going to build a railroad from here over the mountains to Grand Junction. And the way we're going to build it is we're going to start here, and all of us are going to work at the same point and make it grow all the way to Grand Junction. Versus our competitors that are actually going to start it in about 30 places between here and Grand Junction and then just make them meet up. That's what this is doing. Isn't that awesome? Okay, so here we are. We've got a few slides that actually go with a figure in the textbook. So this is what I drew for you just a little while ago on the board, and you can kind of see what's going on. So here we actually have a replication bubble. This, of course, would be in a prokaryote, so the whole thing would circle around. But there's our origins of replication, and leading strand, lagging strands, little tiny wee bits of RNA, and the overall directions they've presented. So that's good. There's a lot of exciting players, and I've given you some of these. So first off, let's just take a look at some of what we see here. So here's the bubble, and here we actually have one strand of DNA and the other strand of DNA. And then here you can start to see some exciting things happen. Now I'm not going to stick on this slide too long, but helicase was on our list of items. We've talked about primers. I didn't even talk about primase. I probably should have. And then here's DNA poly 3. And some good stuff. Lots of 5', 3'. Notice that if this right here is the 3' end of this parental DNA, that means this is the 5'. And notice that the synthesis direction is counter to that. It's opposite of that, 5' to 3'. The last thing I would mention are these single-stranded binding proteins that I didn't really say anything about, but I mentioned their importance, and there they are. So here is a little bit of a blow-up that even brings in DNA ligase and DNA poly 1. I'm not going to spend much time on this, but I did present to you that we've got two DNA polymerases. One of them is called poly 3, one of them is called poly 1. And you'll notice that I presented those in the notes with DNA poly 3 first and DNA poly 1 second, and that's because of the order here. So here we go, page down. Here's the overall picture then. So, as I mentioned, there's actually would be another enzyme out here that I didn't talk about called topoisomerase, but here's helicase, and this is unwinding the helix. It's very important. Here's DNA poly 3, and I should probably get my good notes together here. This primer is under the control of an enzyme that I didn't give you called primase. So here you have helicase, here we have primase. Primase, of course, is controlling the primer, and there's our wee little primer there being formed. I mentioned that the primer length ultimately was 5 to 10 bases long. Here they're actually showing the primer having 5 bases. They're in red. Same thing, 5 bases in the primer. As I mentioned, that could be variable. You could have 5 to 10. And you can see these things starting to form. The leading strand is a big long stretch. So this box here actually is showing the up close details. It's this part right here is what this box is showing. So there's the big leading strand. You don't even get to see the primer. The primer for this one was up here. This leading strand and DNA polymerase 3 is causing this to happen. Single-strand binding proteins. And again, as I mentioned, if you continue into the textbook, you would actually see helicase playing a role here. And then, topoisomerase playing a role here. Here's helicase here. And then all of these then are going to be our growing Okazaki fragments, right? So these are lagging strands. And we're making these things. And you can see now we're actually to the point where we're looking at what ligase does and what DNA polymerase 1 does. So all of those jobs of those substances are actually shown collectively in this slide. And if you get into the textbook version of the slide, it's really nice little kind of blue text boxes coming off of all of these to fill in a few more details. And as I mentioned, there's such extraordinary detail in this text. It's overkill. It's stuff that you can always access and get your hands on, but we need to just understand the basic phenomenon, and that, of course, is that we have, back to here, um, leading strands and then these little fragments, and that ultimately we're going to have to put all of these blue bits together and get rid of all of the red bits and make this into an extraordinarily accurate duplication of each of these strands of DNA. That is the process of DNA replication. Alright, so I think I'm just going to add a little bit of information to this. There are well-established divisions between the people studying prokaryotes and the people studying eukaryotes. The book is going to emphasize prokaryotes. I'm showing you a prokaryotic replication bubble and presenting it in that concept. What I need to do is just kind of prime you into considering the beauty of eukaryote biology. So, in prokaryotes, it seems to be mostly about DNA poly 1 and DNA poly 3. And of course, the model organism for this is E. coli. In eukaryotes, it's much, much more complicated. So far, scientists have discovered 11 DNA polymerases for eukaryotes. And you can read a little bit about that in the book. It is kind of problematic where, you know, you don't know necessarily if it's eukaryote or prokaryote, but right now, safe to say that we're dealing kind of with an easier, more basic kind of prokaryote model that we're dealing with. The other thing that I wanted the other point that I wanted to make is, you know, every one of these things, of course, is a complementary base that's being, you know, inserted to make a double-stranded molecule that has a sugar phosphate backbone. And the errors in completed DNA are remarkably low. The error rate is only about 1 in 10 billion. But originally in pairing, it's 1 in 100,000. So what I'm going to try to say, basically what that means is that you're an A and you're supposed to have a T, but maybe you get a C. And that's an error that happens fairly frequently. But by the time we've completed DNA with all of these enzymes and all of this process, that instead of being 1 in 100,000, those errors are corrected to the point where we only have 1 in 10 billion. We're talking about a mind-boggling quality control system. Mind-boggling quality control system. That's what we've got. Fixing typos. So DNA polymerase, therefore, in this final thing, has this very important role of fix typos, and they do it quite well. Fix the errors, fix the typos in the language. So that's what we've got. Okay, now here's another little eukaryote gem for us. Telomeres. So eukaryotes feature chromosomes have chromosomes with what we call telomeres, and telomeres are non-coding kinds of ends of chromosomes. Now, when I say non-coding, I'm saying that we don't have native genes that live there. Actually, there's some spectacular work that shows that some parasites are moving genes to telomere regions for transcription to make these proteins that they seem to have endless abundance of proteins to change their appearance to our immune system. Radical stuff. But
disregard that little sideline unless you're really a disease-oriented person, because someday you'll take a class in parasitology and it'll knock your socks off.
But back to here, telomeres are generally going to be these non-coding ends of chromosomes. And the idea, I remember one molecular biologist, and this is probably 2 decades ago, describing telomeres as the plastic tips of shoelaces, the protective ends. And that kind of makes sense. They tend to be what we call long base repeats. So I'll say long repeats of one, primarily one short nucleotide sequence. That's what they tend to be, these telomeres. And we have an enzyme for telomeres called telomerase. And